Good evening, ladies and gentlemen. My name's Ian Goodyear. I'm Professor Emeritus at this university in the Department of Psychiatry, and I must apologise because I'm not supposed to be standing here. My colleague, Professor Tamsin Ford, is, but she's been unavoidably detained at a rather serious issue at the clinical school. So this evening we've got a symposium for you. It's within the Cambridge Festival, and it's being delivered by colleagues in Cambridge Neuroscience. Cambridge Neuroscience has over 850 neuroscientists in this university from 30 departments. And this evening we have three of our colleagues very kindly agreed to speak about their work across a range of the neuroscientific disciplines that we have, ranging from human development and early disorders through to computer science, involvement in mental health, and to real tumours in the real brain, which we'll get to in due course. I'm going to introduce each speaker as they come up. Each one will speak for approximately 7 to 8 minutes with their slides, and we'll take questions at the end. So in all your doubtless enthusiasm to ask questions, really would appreciate it if you sort of hang on until our speakers are finished, and then we'll get to the questions in due course. I should also add that this is being streamed, and therefore questions may come from outside this room, and there is an individual here who is gathering those questions via modern technology, and she may well interject, put her hand up and ask a question on behalf of another listener who's not here. So that's the introductions done. It's my pleasure to introduce Dr. Duncan Astle, who is a cognitive brain scientist at the MRC Cognitive and Brain Unit. Duncan is a program leader in work on human development and early atypical disorders in the young person, and he's going to begin our interesting symposium on machine learning and his work in that age range and in those disorders. Duncan.
Ian, thanks very much. I'm going to stand up and walk over to the table. I don't know why I feel more comfortable stood behind the table. Firstly, lovely to see you all in person. And big thanks to Cambridge Neuroscience for putting on the event. It's lovely to be here. So we use multiple different machine learning techniques in our research to tackle different challenges that we face in analyzing data. And I'm just going to give you one example of a way we use machine learning in the next 5 minutes. So who here has heard of Richard Feynman? Okay, so you guys probably know more about him than I do. So I know two things. One is that he won the Nobel Prize for Physics, and the second thing is he said this very helpful quote, which is, "What I cannot create, I do not understand." And the ethos behind it is that if we truly understand the fundamental principles by which something complex happens, then we should be able to recreate that complex thing by reproducing those simple principles. So let's imagine for a moment that I showed you this diagram, and I asked you to recreate it for me. You might think, gosh, that's very complicated. I don't know even where to start. How would I ever get it just right? And the answer is that actually it would be easier than you might think. And that's because that diagram is actually created by a single pendulum swinging and leaving a kind of dribble of paint on the paper. And if you knew the weight of the pendulum, the length of the piece of string, and the force that it began with, then with a bit of physics on the laws of motion and trigonometry, you actually could recreate that complex image. In short, something very, very complex can actually emerge from some quite simple mathematical principles. Well, what about the human brain? So in our lab, we're particularly interested in brain development and how it develops and how it might deviate or diverge in individuals who experience cognitive and behavioral difficulties. And so we are exploring whether or not it's possible to use this kind of approach to think about simulating the formation of the human brain. So how do we do it? Well, we firstly recruit some young people who are usually carefully chosen, and they're, in my experience, very enthusiastic volunteers for our studies. Then they're scanned using various different types of brain imaging. One type of brain imaging we use is called diffusion imaging. So if you imagine for a moment that you had a pipe and you could track the diffusion of water along it, you would find that you would get much stronger diffusion of the water along the pipe than through the wall of the pipe. And so you could imagine that if you could track the diffusion of water, you could infer the presence of the pipe from the way that it moves. And that's essentially how diffusion imaging works. And with it, you can create something like this, which is a diffusion tensor imaging scan, which really captures the major kind of highways in the brain, so kind of thick, kind of fibrous pathways. And then we can use that to create a kind of wiring diagram, a kind of simplified version of each person's brain, which we call a connectome. So each of these spheres corresponds to a particular brain region, and those tubes between them correspond to the strength of the connectivity between those areas. So it's a little bit like a wiring diagram that you might have for a kind of a complex piece of equipment. And so we have one of these for each individual that we've scanned, and then our challenge is to see whether we can simulate the formation of that network over time. Now, I'm not going to get into the maths behind it, but the basic idea is to take some simple sort of mathematical forces that might guide the development of the brain and calibrate them for each person and see how well we can simulate the formation of their network. And to cut a long story short, what we're trying to do is pit two things against each other in the maths. One is that we're trying to force networks to be low cost. So if you're going to form connections between brain areas that are a long way away from each other, just like building a road between two very distant cities, that's going to be a really expensive highway to build. And so there's a kind of metabolic cost to building long connections in the brain. So that's one constraint. And the other constraint is that we want to maximize the overall efficiency of the system. We want to make it easy to get from any one brain area to any other brain area so that information can be easily transferred. So we've got these two opposing forces. One that's driving low cost, and one that's driving high efficiency. And that's essentially what that mathematical equation does. And if you're really into your maths, then those three papers are kind of the foundations for the modeling that we've been doing in this example. And so what you get when you do that, you kind of pit these two things against each other, and gradually in this simulation, you can see that connections start to form. You might be able to see them. It's not a great video, but gradually what's happening is connections are forming one at a time, and those spheres are growing as they get more and more connections. And so kind of playing out over time is the formation of a kind of complex network with a particular calibration of how important cost minimization is and how important efficiency maximization is. And it sounds super duper simple, but actually what we and others have shown is that when you apply these principles, even though there's really only two parts to the equation, you can actually simulate the formation of complex networks like the human brain. And actually it turns out that you can simulate really quite accurately each individual's connectome. So if I took an individual from the audience and we scanned you, if I got the calibration right between these two parts of the equation, I could make a pretty good simulation over time of how that particular network formed. And you might think, well, why would you bother doing that? And here's a few little nuggets of things that we've learnt from this kind of mathematical modeling. So one thing we've learnt is that each child's brain is developing according to a unique set of economic conditions. So this trade-off between minimizing cost whilst maximizing efficiency is subtly different for each individual person. But when you allow that to play out over time, those tiny differences can produce quite diverse networks at the end. So complexity can emerge from quite simple principles. One thing we've shown is that different parts of the equation are probably related to different types of genetic processes. Some to do with kind of metabolic constraints, some to do with things like regulating synapses. What we've done is explore how these simulations work for individuals and for animals who are developing in adverse environments. And what we found is that if an animal is developing in an adverse environment, then there has to be much more variability in where the connections form, much more stochasticity. And you might kind of wonder why that is, and that's an interesting question. And finally, what we've shown is that these simple mechanisms work across multiple different scales. So in this image, it looks like a cartoon, but it's actually a photograph taken of neurons forming a network in a dish. And those little green squares are electrodes, and they allow us to record the activity of the neurons as they form a network. And on the right, you can see the kind of connectome that you can build from the neurons building a network in real time on the dish. And we can simulate the formation of that just like we did with the brains, and it turns out that very similar principles govern the formation of that kind of network as govern the formation of brains. So those are just a few things that we've learned by kind of integrating data from the developing brain with these kinds of machine learning tools. If you're interested, then please do head up to our website, and big thanks to our funders. And I will stop there.
Thank you, Duncan. Well, I hope you're writing down the many questions that has brought to your attention. Very pleased to introduce our second speaker, who's Sarah Morgan, who's a computer scientist, both from the computer laboratory and also from the Department of Psychiatry. Sarah's working on the application of machine learning techniques to understanding mental health conditions. Sarah.
Brilliant, thank you. So I was asked to talk a little bit about what I see as machine learning and how I'm using it in my research. So in terms of what is machine learning, I guess I would describe machine learning as a framework in which we can take some data. So that could be in brain research, that could be brain scans, for example, or genetics data. And obviously we have, at the moment, really increasing amounts of data available to us. Then we also take a model. And of course, again, there are many different types of models that we can use in order to make some sort of prediction. And so that prediction might be, for example, predicting whether or not someone has a particular condition, or predicting how a condition in a particular individual might progress over time, for example. So in my group, we use these sorts of machine learning approaches and other data science approaches to try and predict and understand mental health conditions, often with a particular focus on schizophrenia and other types of psychotic disorders. And really, we're doing that using two main types of data. Firstly, transcribed speech data, and secondly, brain networks derived from magnetic resonance imaging data. Similar to what Duncan's already described, but really here what we're doing is looking at sort of macroscopic connectivity between fairly large-scale brain regions. So that's a sort of very broad overview. I thought I would just pick out a couple of particular projects that we're working on, just to give you a bit more of a concrete idea of the kind of things that we do. So one example of some work we're doing at the moment is trying to use computational markers of patients' speech in order to try and predict relapse for patients with psychotic disorders. So our work has shown that these sort of computational markers of speech that we can extract with different machine learning methods, for the experts, sort of natural language processing markers is really what we're using here. And what we found is that these kind of computational markers of transcribed speech really show quite large differences between patients with schizophrenia, for example, and healthy control subjects. And at the moment now what we're trying to do is to collect some more data here in Cambridge, which is really from patients who are trying to come off medication and who are at some risk of relapsing. And what we're keen to do is see whether or not these computational speech markers can then predict which people are going to relapse, and potentially also to sort of monitor that relapse and maybe send sort of early information back to the clinician that maybe this is someone that they need to pay particular attention to. So that's an example of the work that we're doing on speech data. As I say, we also work on brain connectivity. So, for example, some of the work that we've been doing has been using machine learning to try and identify patterns of brain connectivity that are particularly relevant to schizophrenia. So here, the blue regions are where we tend to see reduced brain connectivity in patients with schizophrenia, and red regions are where we tend to see increased brain connectivity. And then another way that we've been able to use machine learning is then to take those brain maps and relate them to gene expression data. And that's particularly exciting to us because brain MRI is fantastic because it gives you this sort of non-invasive window into the brain, but it is also sort of millimeter scale resolution. And so it can be a little bit hard sometimes to really sort of get down to the sort of underlying biological mechanisms that happening at much more at the microscale. And so this is sort of one approach that we've been taking, is to relate the brain MRI to gene expression data. And in this particular project, that was able then to tell us some information about what sort of genes seem to be most implicated. And we think that this might be interesting as sort of potentially highlighting new targets that could be used for drugs. So I thought I'd just finish by outlining what I see as some of the main opportunities and some of the main challenges for machine learning in brain research. So I think in my work, I see two main opportunities. One is using machine learning really to try and predict individual disease trajectories, to give additional information to clinicians to help inform their diagnoses and treatment. And then secondly, I think there's also an opportunity for machine learning to help us to better understand the brain in health and disease. And of course, that can then potentially lead to sort of new therapeutics with that increased understanding. In terms of challenges, I think for me there are sort of three main challenges that I think are particularly interesting. Firstly, there are still technical challenges. Obviously, there's lots of research ongoing into machine learning. And I think that as we start to use machine learning more in applications in the brain, in healthcare more generally also, that's sort of bringing up more or putting more focus on particular technical challenges. So for example, how to make algorithms more explainable, which is very much an open machine learning research question at the moment, and also how to incorporate our prior knowledge of the brain and of mental health conditions into these sorts of algorithms. There are also ethical challenges. I think those are really around how to ensure machine learning is used in ways that really benefit patients. And then finally, I think there are some really interesting educational challenges. I hope that it's already becoming a little bit clear that this is a really highly interdisciplinary field, drawing together people from computer science, engineering, physics, but also clinicians, biologists, geneticists. And I think we're still learning really how to foster those conversations between these different fields. So thank you very much.
Thank you, Sarah. Our third speaker is Stephen Price, who comes from Clinical Neurosciences Department at Addenbrooke's. Stephen is a neurosurgeon, and he works with his group on slow-growing tumours, and he's interested in how can we predict trajectory, and how can this help him and his group help all of us to understand best treatments for individuals with these extremely difficult conditions. Thank you, Steve.
Thank you. And thank you again, Cambridge Neuroscience, for giving me the chance to discuss some of the work we're doing for the clinical school. So I, as I said, is a neurosurgeon, and one of the things we do is we look in our clinic is we have a large number of patients who have been found to have slow-growing tumours that are under follow-up, and we're following them up over a long period of time. And it's the whole thing about trying to predict what's going to happen to these patients is the difficult part for us. So what I thought I would do is I would show two examples of two patients. These are two real patients. I may have changed some of the details, but these are two real patients. So the first patient is a man in his 50s who has a bit of dizziness and goes and sees his doctor, gets referred to the hospital, and as part of his investigation for his dizziness, someone does an MRI scan. And a very astute radiologist picks up a problem. Anyone see the problem? It's there. Okay, there's a little thing there. And so he's called back, he's told, well, there's a problem with your scan. He's called back and he's given another scan, this time with given an injection of contrast. And this lesion shows up very, very, very well there. And this is a tumour within the lining of the brain, so-called a meningioma. These are the commonest tumours we see. For every 1,000 patients who have an MRI scan, we pick up 9 of these. That's how common they are. And in our weekly MDT meeting where we discuss new patients, we have our meetings tomorrow morning, I can guarantee there's going to be 2 or 3 of these patients that we will pick up this week, just for how you manage it. Now, we know most of these patients, the tumour doesn't grow. But we follow them up. And we've been following this man up for a few years now. And after a couple of years where it didn't seem to grow very much, you can see that as time goes on, this is getting bigger over time. He's completely well, has absolutely no symptoms, no problems whatsoever. And we are following this up at the moment. And it's the question of when is it we should be intervening? When should we be thinking about doing an operation to remove this? When is the best time to do that? And part of the information we would love to know is how fast is this growing? Is the growth rate steady or is it getting faster? And also for the other groups of patients we have, it will be really useful to know, can we identify that 90% that don't need following up because it's never going to grow, and separate and follow them up less intensively, and also pick up that group of patients where it is going to grow, and we do need to follow them up in, more intensively over time. And this is what we don't have at the moment, and this is one of the tools we're trying to develop using machine learning to predict. Second patient. This is a man in his 30s who had a fit. Out of the blue, fit and well, runs marathons, just had a fit one day. Brought into hospital and has a scan and is found to have this. And this is a tumour that when we give the injection of contrast, it doesn't light up. It's within the brain and it has the appearance of what we call a low-grade glioma. And this is a slow-growing but malignant tumour of the brain that will grow very slowly for a period of time and then at some stage may grow faster. And it's trying to sort of follow that up is very difficult. So here's when he had his fit, and after we then brought him in and operated and removed pretty much most of the tumour. It's very hard to see a lot of tumour left behind. It's very hard to see on these images. So then he comes for follow-up, and we're following him up. And he gets followed up on a regular basis. Nothing much happened between those two scans. Is there any change in these scans? It's very hard to say. What about now? Has that changed? Possibly. There's a little bit of, you can see the bit of the white area by the cavity, the hole where we've taken the tumour out. And again, slow changes we're seeing over time with this tumour. He asks us how fast it's growing. I don't know how do we tell him? How do we say how fast this is growing? It's very difficult. The way we liken it to is giving someone a flower and handing someone a flower, handing them a ruler and say, measure that flower. It's really hard to work out how big this tumour is and how fast it's growing. And then between November 2020 and January 2021, this looks completely different. And this is another problem we have, is that between these two time points, the MRI scanner changed its sequences. They changed everything around. New upgrade, new sequences. Now, how do we compare these two sequences? Because they are different. They are different from what we had before. And again, we've been following up over time, and slowly this has been growing over time. And in fact, we operated on him again fairly recently to remove the remaining part of his tumour. One of the other problems we have is that we now can get images, not only just seeing them in a single plane, but we can see them in different planes. This is why our images change, because we could then reconstruct them in any way we want to. And so you can see here, if you looked at that part here, you'd see a very small amount of tumour. If you look further down, there's more tumour there. And the other problem we have is not only do we get this when we scan patients, but we get all of these images as well. And each of them tell you very slightly different things. And trying to get a radiologist to look at these and identify very subtle changes is really difficult. And this is where machine learning really comes into its own, to identify these changes, give us an idea of changes in size, of how fast these tumours are growing. Some of the simple things we're trying to do is even just to outline where the tumour is and measure the volume and see how the volume changes over time, but also using it as a predictive tool to then say, this is somebody we're worried about, or this is somebody we're not so worried about. We can then scan them less frequently. Okay, thank you.
Thank you very much, Stephen. Well, those are our three speakers, as you have appreciated, from very different realms of our Cambridge neuroscience community. So I get the prerogative on behalf of the other 874 neuroscientists, most of whom are probably not here, some might be, is to, someone's nodding, no doubt others are too, is to just put three questions, one each, to you, not specific questions. The first, with something that we've all discussed, the brain. 100 billion neurons, 100 trillion minimal interactions. Are we really seriously hoping in some way to model this organ in a coherent manner? That's cross-sectional. And I'll leave that question with Duncan to give us a great insight. The second question to Sarah is predicting treatments in an organ that appears to have more non-linear than linear relationships with itself. And you've told us about the massive individual differences that there are. Are we really going to get machine learning to help us with treatment at the individual, personalised level? What's your view? And then to our neurosurgical colleague, is this the end of radiologists, Stephen? Are we going to end up with a machine greeting the patient coming in, robot X, do take a seat, and then pumping out the results without any human beings needed at all? So those are the three questions from myself on behalf of everybody else. Let's start with Duncan.
Great, thank you. So I think a really key thing to think about is when we say study the brain, there are so many different ways that we could do it. And there are so many different scales at which we could do it, right? So the stuff that I showed, like building networks, we often model the brain as, say, 400 areas that are all may or may not be connected with each other with varying strengths, right? So that's a really simplified model of the human brain. I think Sarah used the word like macroscopic, right? So that's what we would call the macroscopic level, right? Then the other extreme, we could take some brain cells and we could grow them in a dish and we could observe the formation of a tiny network forming in real time. At the minute, I think you have to choose your level of analysis. You have to choose where on that scale you want to situate the analysis you're doing. But one of the things that I really like about machine learning is that it can provide us with a kind of mathematical framework that we can use to work along that scale. So, I can apply similar models to my simplified, 400 by 400 model of a child's brain, and I can apply the same modeling to the formation of a network in a dish, and I can explore whether similar principles operate across those different scales. So at the minute, it's certainly not the case that we're kind of capturing the full complexity of the brain. We're having to choose where it is that we situate the research. But one of the exciting applications of machine learning is that it provides us with a kind of framework that would allow us to integrate those in terms of the kind of mathematical principles that govern them.
Thank you, Duncan. It's a great answer to an impossible question. Sarah.
So I guess in terms of clinical translation, as I said, I see there really as being two routes to clinical impact. One being to better understand how the brain works, potentially leading to new therapeutics. I see a lot of the work that I do on brain imaging as being sort of in that direction, and I think that's a longer burn in terms of actually sort of translating that to the clinic. The speech work that we're doing, I think, is potentially interesting from a different angle in terms of actually being able to sort of predict individual disease trajectories. And there are some quite strong signals there, particularly for schizophrenia, which is where schizophrenia has long been known to often involve sort of changes in language. And so there do seem to be quite strong signals there. And so that's something that we're looking at as potentially being a bit closer to clinical translation. But definitely there's a lot more research to do in either direction.
So we're going to keep scientists employed for a while then, is that? Yeah.
I think so.
Good. Poor old radiologists and radiographers.
So I think we're going to keep them employed as well for a while. You can never remove the radiologist from the equation, because the thing about machine learning is it'll tell you, first of all, if you say to it, this is a meningioma, for example, it doesn't question that, whereas the radiologists do, and they'll look at it and go, well, actually, no, I'm not happy with this. There's something else going on. Or, so for example, that scan, the first scan I showed you, I don't know whether you would have ever been able to train a machine learning algorithm to detect something as subtle as that, but a radiologist looking at it went, what's that? That's a bit odd. And then, so I think that's the first thing. What these tools are going to do is provide new information for the radiologist to do their job better, I think is the answer to that. Is if we can give volumes and show volume trajectories, they can put that into their reports, and that will help us with them reporting on this. We can then have that to help our managing patients.
So this is machine helping man.
Yes. And in fact, the approach we've taken is always has what was referred to as a human in the loop within it. So the things that will provide information for someone else to look at and check and make sure they're happy with it. And then we can then use that to make decisions from that.
Thank you very much. Well, I think I've used up my prerogatives. Let's have some questions from the audience. The gentleman's just shot his hand up there. I think, yes. You, sir, yes.
Hi, I'm going to cheat a bit. I've actually got two questions, if that's all right. One for Duncan. You said that maths is in the papers, but could you give a sort of layman's description of what the algorithm is for simulating the brain getting built up? And for Sarah, the question is, how's the gene expression data that you sort of mentioned collected, and how is it mapped back to the MRI data?
Shall I go first? So the equation has two components to it. So the first one is the costs term, and you could approximate that as something like the Euclidean distance, or literally physically how far two things away are from each other. You could actually, depending on the type of scan you have, you could use the length of the fiber that connects those two areas as your distance term, right? So we approximate cost as the distance between two brain regions. And we trade that off against what we call the values term. Now, the values term could be any characteristic of those brain areas that's not to do with their physical location. So, for example, something like their neighborhoods. So maybe we've got two brain areas, they're a long way away from each other, but they share lots and lots of neighbors. And so that would give it a high value term, and then we trade those two things off against each other. And essentially that's how the equation works. And then for each individual simulation, we can calibrate the relative contribution of those two parts of the equation. And then I've given you one example of a kind of values term, but you could have multiple different types of topological term, or different types of characteristics of the brain areas. And what you're essentially doing is repeating it multiple times and seeing which properties do I need to give the equation in order to simulate something that looks like a real brain network.
So you're building it up iteratively, taking the cheapest or most productive connection each time that would improve the network.
Yeah, exactly right. So if I was simulating your brain, I would maybe run it 10,000 times, and each time I would either up or down weight the relative contribution of each part of that equation, right? So for each simulation, the calibration is slightly different. And so in the end, I have sort of 10,000 simulations of your brain, and then I can look through the data and say, right, which of them produces a brain that looks exactly like yours? And it turns out that some really common principles govern everybody's brains. And that's the basics of how it works.
That's excellent, thanks.
And to answer your other question, so I've glossed over a lot of the details there, but essentially what we're trying to do, as I said, is to link this very macroscopic brain MRI data back to something, back to the gene expression data. So what we do is we take the brain signal that we're interested in. So in the case that I described, that's the sort of case control difference in brain MRI in schizophrenia compared to healthy control subjects. And then the way that we're linking that to the gene expression data is to look for sort of combinations of gene expression for different genes that can match that brain MRI signal. And the subtle thing is that the gene expression data actually comes from a group of healthy individuals, because the gene expression data has to be obtained post-mortem and is obtained sort of across the whole of the brain. So this is the data that we're using comes from the Allen Human Brain Atlas. It's sort of an openly available data set, which is being used by quite a lot of scientists around the world now. but it's quite a small data set of this post-mortem gene expression data. So it's not gene expression data from the patients with schizophrenia that we got the brain MRI signal from in the first place. But I think that's a sort of general theme for a lot of these machine learning approaches at the moment, is that, as Duncan was saying, they allow you to combine different types of data and data at different scales. And that seems to be one of the really powerful things about them.
It is.
Amazing, thank you.
Thank you very much. I'm looking around for more hands. Sir. Yes, you, sir, and they're coming rapidly with a microphone.
Thank you very much. I've got quite a loud voice anyway, Duncan, actually. Biological systems are inherently variable. I know that from my own research work on cardiospectral analysis. How reliable, if you did the same sequencing on the same individual, but on different days and in a different environment, how reproducible do you think the data would be?
Do you mean from the modeling perspective or from the scan perspective?
The scan perspective, one would hope, would be fairly real. It's the modeling perspective.
The scan does change from day to day. It depends on what you mean by. So the example I gave was using diffusion imaging data to calculate connectivity, right? So we're looking for the presence of physical tracts in the brain, and that's pretty reliable. The other approach would be to measure metabolic or deoxygenation in those brain areas using the fMRI and correlate it over time as a measure of connectivity. And my experience is that, at least in kids, that doesn't tend to be as reliable because there's all sorts of issues with how you calculate it, and if a kid moves ever so slightly, then that can... Anyway, so the scan, depending on the measure you use, should be pretty reliable. So the modeling, so I have repeated it, say, on the same individuals that came back 2 years later, and you can see that it is replicable because there are changes. Now, that could be because...
Child's development.
Exactly right. We don't know whether that's the inconsistency in the modeling, but we've got some individuals where we've got multiple scans at this, at very close in time on the same individuals, and you do get really reliable results. And then they do tend to be good predictors of other things, for example, cognitive performance. So the way that someone's connectome is organized, at least in our data, gives you a really good prediction of overall cognitive ability. If someone's got cognitive difficulties, for example, you can tend to predict it pretty well from the organization of their connectome, which is one of the reasons I quite like it, because that makes good sense to me.
Thanks very much. Just incidentally, we are using cardiac MRI scanning by machine learning, and actually the initial data suggests it's just as good as an experienced radiologist, and better than most, well, less experienced radiologists.
Right. Moving on.
Could we take an online question, if that's all right?
Where are
down
you?
here.
Oh, you're
Sorry.
Yep.
So it's not directed at anyone in particular, but I suspect it's more for Sarah. Could MRI scans become a standard diagnostic procedure for mental health conditions, or is machine analysis of patient behaviour a more likely approach?
So I guess, as I say, most of my work focuses on schizophrenia. With schizophrenia, we can use brain MRI images to distinguish patients with schizophrenia from healthy controls with about 80% accuracy. That seems to be sort of where we get to with that from lots of different groups worldwide and lots of different methods. It's also not a particularly clinically interesting question. And so what clinicians tend to be more interested in, certainly as far as schizophrenia is concerned, is trying to predict outcome for individual patients. So I think at the moment, and with my work, that's sort of more the direction that we're trying to push in. But I think the signals are relatively weak, to be honest. And so I think really it's more about using brain MRI to build understanding. The other issue, of course, with trying to use machine learning to just sort of get basic clinical diagnoses is that, schizophrenia, for example, is extremely heterogeneous. There's ongoing debate about sort of what schizophrenia really is and if it should really be a number of different disorders. And so really, in some ways, you want the machine learning to challenge some of that and to kind of move us forward in terms of our understanding. And so another thing that you can do with machine learning is look, for example, for subgroups of patients who maybe have different biological mechanisms underpinning a particular disorder. So I think I see machine learning more as being used for those sorts of challenges. And I think we're certainly a long way off using machine learning in the clinic in that sort of way.
Just to add to that, I do think that using machine learning for the behavioural phenotyping is going to change our construct of what a clinical diagnosis is. And certainly in the work that we did, of course my group carries on perfectly well without me, is that you do get different connective patterns that are strikingly previously non-emerged in other techniques. So I'm sort of a bit more hopeful about machine learning giving us some degree of better behavioural phenotyping precision. I agree with you about the brain issues. There's a long haul there for that, isn't there? Lady there, yes. No, yes, you. Yes, you. You need the microphone, and it's coming.
This is a question for Duncan. I was wondering, applied to developmental psychology, how might you use mathematical models to simulate not neural changes, but like, say, conceptual changes? For instance, when children develop, from basic numerical intuitions of 2 is greater than 1, develop from that more sophisticated abstract concepts of natural numbers, because these conceptual changes don't have any clear neural correlates that you can identify from imaging that you can compare your model against. And like a slightly secondary question is, say you're a developmental psychologist who's not a machine learning specialist, how might you improve your understanding in this area to be able to use these models in your own research?
Great, 2 great questions. So the first one is, I don't know how much this, at least as I've given the example that I've given, I'm not sure how much that would tell you about those sort of big conceptual leaps forward that kids make around certain kind of milestones. But that I still think that machine learning can potentially tell us something about those, but just probably not through the use of neural data. So another thing that we do, which has sort of been touched on already, is applying machine learning to different types of data that you can collect from an individual, whether it be kind of standard cognitive assessments or wearable technologies or anything that really captures the behavior that the individual displays in everyday life. And one of the challenges with kind of developmental conditions such that we're interested in is that they don't tend to fit into neat categories. And so even when someone has a formal diagnostic label, that's not always a tremendously reliable indicator of their specific difficulties. And so what we do is often take large data sets, large mixed heterogeneous data sets from lots and lots of kids, and see whether the machine learning algorithms can identify, as Sarah was saying, kind of groups of individuals. And you can imagine that you could do the same thing across time. Can I detect moments in time when particular changes seem to occur in a kind of data-driven way? So I think that it could tell us something about those sorts of shifts in thinking, provided that we can gather data that we think would reflect them. But I don't think it necessarily needs to involve the brain. And your second question was about, how does one get into machine learning? That's a really good question. There are some nice review papers out there in things like Trends in Cognitive Sciences that are nice to read and make generally good sense. That's certainly where I started. And then the next thing I think is to try some very simple things in simple data sets, often data sets that you may have made up through a simple simulation. And that's how I sort of learned how to use various different kind of bits of code and gradually sort of built from there onwards, I would say. So sort of some conceptual underpinnings and then sort of try your hand at it with some very simple sets of data that you may have made yourself.
So these models that you just spoke of...
Can you use the mic? Thank you.
The models that you just mentioned, would they be descriptive and in that they will predict when, at what age certain conceptual changes take place, or would they be truly kind of explanatory of what kind of mechanisms actually are driving these changes?
That's a good question. So the example that I gave of the kind of generative networks, I would argue that to some extent is explanatory, is because you can then unpack the simulation and explore, given the properties that we observed at the level of the whole system, how do they emerge from the simple building blocks that we gave it? The example that I gave you about using machine learning as a way of kind of breaking down complex behaviors and observing particular changes in time, I think as I've described that would be more about optimally capturing when those happen and how they vary across individuals. So at that stage, I don't think that's explanatory, but capturing them properly is the first step to properly explaining them. So once we've got those sorts of things, that's often what we use, for example, rather than, say, a formal diagnostic label. We often use the outcome of a machine learning algorithm that's captured those differences more optimally. Even though they themselves are not explanatory, I think that's kind of the first step along the journey to finding something that is.
It also suggests, don't you think, that by using machine learning, we might be able to deal with the incorrect explanations that have stemmed from the incorrect phenotype that we've been living with, whether it's A, B, or C conditions.
Yeah, and what you often observe when you get these big data sets is that even though children may have the same diagnostic label, there's lots of variability which the machine learning is learning about. And you can soon see that internationally, people are recruiting different samples, all of which have the label, but that are quite different from each other. And that explains lots of the inconsistencies that you see.
Oh dear, don't tell me about that. Don't we know when it comes to collaborations like that? Yes, yes. Anyone from the back who's obviously not fallen asleep is desperate to ask a question.
This lady has been waiting for a little while, if that's all right.
Yes, do.
Hello, this is for Duncan, really. Have you seen any major differences between, say, groups of children that are diagnosed with autism, ADHD, dyspraxia, dyslexia, where there are a lot of overlap of symptoms? And are there any relationships with the sort of cognitive tests? Have you seen any relationships, whether it's WISC tests or other types of tests that are used for diagnosis of these conditions?
Yeah, so there's one study that we conducted as part of a collaboration called the Centre for Attention, Learning and Memory, or CALM. And the idea of CALM was to collect this large data set of all comers, so multiple practitioners in educational clinical services could refer children to it, and they would visit us for the day, and we would give them sort of 3 or 4 hours of cognitive assessments, multiple behavioral questionnaires, and of course we would record any formal diagnosis if they had one, or multiple. And then we used machine learning to see whether there are different groups of individuals within the data set. And we found that there were different groups of kids with different types of cognitive difficulties. Some more akin to kind of more language-like difficulties. So parents would spot they had kind of syntactic difficulties, structuring sentences. They also had various different kind of cognitive difficulties involving verbal information. And then other groups of kids with different types of difficulties. And then, of course, what we can do is test whether what the machine learning has learned maps on neatly to the formal label that the kid came with. And the answer quite clearly was that they don't map on well, which you may or may not have predicted, but anyway. But that you could distinguish those individuals in the way that their brains were organized. So connectomics in different brain areas seem to be more predictive of being a member of these sort of different subgroups with different cognitive difficulties, which suggests to me that sort of underneath the formal kind of label are a set of difficulties that different people encounter that are themselves probably more closely related to the neurobiology of the brain.
Would you say that that's probably connected with the fact that the way these conditions are often diagnosed, it's quite primitive? It's sort of on symptoms, it's a little bit like, you know, in the past where people had stomachache and it actually could have been caused by all sorts of things that that what you're doing will actually be able to identify something at a more basic level.
I think there's all sorts of reasons why there's that sort of mismatch. And I think one of the reasons is because, at least in the UK at the minute with the current system, it's incredibly difficult to get to see the kind of clinical or educational support that you need. And that then means that the formal labels that people have can sometimes be haphazard because there are undiagnosed difficulties that they're experiencing, and the diagnosis may reflect the person that they managed to get to see. And so there's all of that in there as well. So I think actually the diagnosis itself is still a major landmark moment for the children and the families that should leave for additional support. But the problem is getting to that point can be so challenging sometimes that using it as the formal label itself can sometimes be a bit haphazard.
Yep, you.
Okay, great. Thanks so much. I have a question for Stephen, and it's kind of a multiple kind of part question, but I think it's pretty straightforward. I'm just kind of trying to put myself in the place of being told in a hospital that I have a brain tumour and that a machine is helping the radiologist or the brain surgeon decide whether or not I'm going to have it removed. Because in my mind, and this is probably too much Grey's Anatomy on my part, but I kind of thought, brain tumour equals bad, remove it before it spreads. And I know that you're tracking it, but is it not always better to remove it when it's small? And my question kind of around that is, A, what do patients think when you talk to them? Because presumably you tell them that they're in a study or something, that you're trying to kind of use this different way of learning. And what was my second part of it? That's my COVID brain now. I'm trying to think of my second part. But is it, and the whole thing about, oh yes, this is the other part. Is one of the reasons why you would be doing this, and this might be the other cynical part of me coming out, is it a kind of a cost thing, cost-benefit analysis in terms of financial? Because we can't, I couldn't believe when you said those tumours were so common. Are they all malignant, or is it a cost thing in terms of the NHS that we can't operate on everything until it's at a particular point? Yeah, you kind of get my question.
Yeah.
Thanks.
So to answer your first question about the operating and choosing a time for operating, we know that so this is really focusing really on the meningiomas, these often incidentally found meningiomas. Well, we know now that the majority of these will never grow. They seem to go to a certain size, so you see them on the scan, and then you follow them up for years and years and years. We've got patients we now follow up for sort of 10, 15 years, and they haven't changed since then. And the person has no symptoms at all from it.
They're not malignant?
They're not malignant. They are benign tumours of the lining of the brain. But we know some do grow, and that's the problem. There is always this risk-benefit from operating. There's a saying in neurosurgery that you ain't never the same when the air hits your brain. So you always have to be a little careful about when you choose to operate. It's trying to operate on somebody where you're not going to make them any better. You put them at risk, and all you're doing it for is to perhaps, if this grows, but we don't know if it's going to grow or how fast it's going to grow or when's the right time to do it. So the first example I gave you, the first patient, he's very happy to keep on watching that and see what's going on. The second part of your question about NHS resources is partly yes. This is becoming an increasingly common problem that we're finding these tumours, and following up these patients regularly has got implications. And radiology departments have a finite resource in terms of how many scans they can do, and if they keep on doing these ones, we miss some of the other ones. If a scan and you're coming up to your scan, that's all you think about. And until someone says to you, it's fine, there's no change, that is all you think about. And it just takes over your life. And there are a lot of people who get these really bad problems with anxiety around the time of their scans that just completely takes over their life and doesn't really have a chance and they would be very happy not to be followed up on such a regular basis. And it's the two balances. But there is a large part of it's going to be is cost. This is an increasing cost on the health service, just following up these incidentally found tumours that may never cause a problem.
Stephen, I think you just added a sub-diagnosis to the DSM classification, clinical taxonomies, scan anxiety.
Anxiety, yes.
Not scan. Having experienced that, even doing multiple research scans, I'm not surprised, but I shall check out with the other psychiatrists if we've now got a new disorder to add to all the other ones that we don't understand. Do we have more? There's a, yes, down there on the front row.
All right, thank you for your time. I was going to ask, how do you standardize machine learning algorithms across various different machines and the different lenses that the radiographers will use in different clinics? And how do we improve public confidence in machines aiding in this diagnosis?
Should I take that? Because that's probably more related to my work. And this is a real big issue. So one of the big problems with a lot of work being done on with MRI scans is that every hospital has a different there's different manufacturers of scanners, and each scanner does things very slightly differently and produces a very slightly different output from it. And some of the scanners don't even tell you how they do it. They have these fancy little things that make the scans a bit brighter or a bit less bright. And as you saw the example I gave, that every now and again someone changes the sequence and it changes everything. You then suddenly have a new sequence to deal with. And that is really difficult. The only way to do that is collecting a large amount of data from lots of different centres, if possible, that have used different scanners, and patients have been scanned on different scanners. And that's what we've been trying to collect, trying to collect this database of scanners that we can actually do this research on. The problem we have, though, is that this is okay, the numbers are fairly common, but if you think about the Cambridge Brain Tumour Clinic, we are following up about 600 patients at the moment. There's patients elsewhere and other groups where have been followed up, and that is for the whole of East of England, whole of East Anglia. So there are not vast amounts of people out there for us to study with that. The second part of your question is a really important point, and that is to do with how people will take the results of a machine learning algorithm. But a computer says this, and whether it's going to change your management. And if you don't mind, I'm going to ask a question to the audience, if that's all right. How many of you would just put your hand up if this is true? How many of you would be happy with a machine learning algorithm once it's been trained and we can show that it's working very well, and we then will look at it in an independent data set to prove that it's doing a good job, that tells you that your tumour is not going to grow and we don't need to follow you up anymore? How many people will be happy to not be followed up? So I would say, what, a third of the audience, would you say? About a third of the audience have put their hand up for that. So there's a lot of people there, 2/3 of the audience aren't happy with that. And this is an issue, and this is a piece of work that we are looking at doing, which is to explore this how to make it acceptable to patients. And I think sort of touched upon this thing about explainable AI, where you actually understand, because most of the time for AI is you have a lot of data, you say this is one type of this is a group of tumours that grow, this is a group of tumours that don't grow. You put it into some sort of black box and it gives you an answer, and you have no idea what it's done in the middle of it. But if you can find ways of actually understanding the principles, what is it he's picked, what is it they've picked out, what are the important parts, I think that might make it a bit more acceptable to individuals.
Do we have more hands? Gentleman's had one question. I'm happy to come back. Is there one from... Online. Okay, one online. Yes?
Sorry, I'll give it back to you in a second.
So another online, it's a sort of series here. It says, with my schizophrenia diagnosis, I'm not sure about this. As a programmer, what about data protection? And could scans be used to make decisions about potential? And I think that means sort of human potential in general.
So I think, as I said, I think there are lots of questions around the sort of ethics. Definitely having, definitely it's really important to involve people with lived experience. I work quite closely with a group of people with lived experience. Generally, often the concerns actually that so this is a slightly different point, but generally a lot of the concerns that they've raised have been sort of things that people have been talking about a little bit earlier around sort of cost efficiency, and is this a way of making things cheaper? And actually that they really value the discussions that they have with their clinicians and the therapeutic relationships. And so I think it's absolutely right, as you were saying earlier, that this sort of information and machine learning approach is a way of giving additional information to clinicians. It's not about the machine sort of making decisions. And in terms of the second point, I think definitely these things need to be conversations around what we think machine learning should be used for and should not be used for. But actually, I mean, that often is sort of ethics questions that we're quite used to in research already. In some sense, the fact that it's machine learning doesn't sort of change the fact that you still have to go through the same ethics boards, and I think that's absolutely right.
There's also an ethical dimension in using the data. So I've had participants say, our data are our story, and we want that story told. So there's the ethics of, one thing we're always worried about is identification, is that you could use an algorithm to identify who an individual is within a data set when it's meant to be fully anonymous. But then the flip side of that and the kind of counter pressure is that I think there is also an ethical obligation to make sure that people who have given their data and taken part in the study, I think there is also an ethical obligation to make sure that the benefits of that data are maximized upon. And so, as Sarah was saying, finding the best way of doing that is sort of where the trade-off sits.
Did you have another online question?
We do have a few online.
We'll take one more from you, and then we'll see if there's anyone in the audience who's got more things to.
I think it's probably for Sarah again, but please feel free to chip in. What sort of behaviours other than speech are likely to be useful for machine learning analysis of mental health conditions?
So what other modalities?
What other behaviours other than speech are likely to be useful?
So people look at lots of things. I know some people at the Turing Institute, for example, who are working on gait analysis, sort of movements. I know there's some discussion about looking more at sort of facial expressions. Going back again to the ethics, obviously that can be difficult. Actually, in some sense, the brain MRI is quite hard to identify someone from a brain MRI image, particularly if the face has been taken off of the image. Whereas speech data, for example, is much easier to recognize someone from. And similarly, obviously, like facial expressions and so on. So it raises a number of these other questions. But definitely people are looking at lots of different types of data. Are there any other main ones that I've missed?
I guess one of the big pushes is to collect data from people's mobile phones. So about where they go, the kind of usage of the phone. They often try and then put an app on it, such that at multiple times you're asked sort of questions about well-being and mental health, for example. So that you get these really, really rich data sets that are a scale that you would never be able to achieve in more conventional means, and you get this kind of rapid sampling of experience and behavior. And so that's one area where people are applying machine learning to those kinds of large data sets gathered basically from the data on people's phones.
Did you want to ask another question? Do we have a...
Sorry, it's just a comment.
Oh.
When you asked the question about whether people were happy about machine learning and following up, you just gave this binary choice, either follow up or not follow up. And it may be that more people would be happy if you had asked, would you be prepared to have longer intervals rather than not be followed up? And I suspect that people might be more prepared to have a longer interval rather than not be followed up.
That's an interesting point. Let's try that. Who would prefer to have longer intervals? Instead of having, say, 6 monthly scans, may go to sort of year, 2 years. That's more really most of the audience, I would say.
let's go back to the gentleman who had another question, the biologist here.
Thank you very much, Stephen. The incidentaloma, which is what we call them, is a nightmare because there are a number of people now going through scans, well people scans, who tend to be a little more anxious perhaps than others, so scan anxiety is a real problem. What would you tell people about driving? Because with a meningioma, I understand, there is a potential you can fit, and therefore you have to let the DVLA know. Is that not right? Would you then stop them driving?
So the DVLA have got quite strict rules on this, and our answer is you can drive, and you don't. For an incidentally found one, I don't even think you need to notify them about it.
Okay. Would you stop them if it started to increase in size?
Yeah, so that's when you would have to start thinking about informing the DVLA when it starts increasing in size. And certainly if you operate, one of the problems with operating, as soon as you operate on someone, they can't drive for at least 6 months afterwards.
Thank you.
A gentleman over there with his hand up, I don't know, a dozen rows back. Good exercise for the microphone holders.
Are there any concerns about a loss of radiologist acuity with an increase in the number of routine scans which are read by machine?
In terms of having lots of normal ones to see and picking up ones here, I don't know is the honest answer to that. I think one method that has been suggested in terms of how you might use a diagnostic MR capability using a machine learning approach to it is if you turn the sensitivity up so it's very high, it might pick up and will pick up a lot of things that probably of no real benefit, real importance or concern, but it might allow them to triage which order. So if they're given, say, 100 scans to look at, the top, say, 30 might be the ones that the algorithm said, we're a bit worried about these, can you have a proper look at these? So it might actually in some ways help them to concentrate their mind on the ones that are seeing. And I think there's talk about whether we could, I don't think there'll ever be a day where we take away all their work from them and say, no, there's no need to do it. I think that's where it will come in, we can triage things and improve the triage of scans for them to look at.
Over there, you want to pass it on? Thanks.
When you say machine learning, do you mean, is it more of just a way to crunch complex data, or is it actually an algorithm which will learn and, not learn, but like, will it get better over time by giving more weight to certain factors? And if so, how would you mitigate a risk of it giving too much weight to certain factors? For example, when machine learning was being used for job interviews, some of them gave unnecessary weight to people having books in their background or something.
I'll have a go. So I think in the machine learning world, there are some purists who would say machine learning equals a specific set of algorithms, and if you're not using those, it ain't machine learning. But I think a really helpful way of thinking about it is that there's sort of two broad sort of ways of using machine learning, sort of like what we call supervised, which is where you've got a particular outcome that you're trying to predict. And whether it be a kind of clinical diagnosis or a specific outcome that is known. And so what you're essentially doing is exploring whether or not there's information in your data set that can predict that outcome well. So the other type of application is the unsupervised, which is where essentially you're not trying to predict any specific outcome, but you're trying to sort of optimally capture a data set by learning about it. Now, both of those involve learning, right? So one is about learning to represent information, often kind of complex information in a kind of more simple format. And then, whereas the former is about learning to predict a specific outcome. And the problem with both of those is you can get what's called overfitting, right? So in addition to the meaningful signal that's in those big data sets, there'll be lots of noise, right? Lots of stuff that's not of interest. And the risk is that the algorithm starts to pick up and represent that information, or start to pick up and use to predict some outcome, uses that information. And the cast iron test, or whether that is happening, is whether you get generalizability, right? So if you took your algorithm, you trained up, it had learned to represent data or learn to predict an outcome given certain characteristics, and then you try it on a new data set that wasn't involved in the training. And if you've learned something genuine, then the algorithm should work in that novel data set as well. And if it doesn't, then that suggests that your initial result may just have been, learning about the books, like overfitting to the wrong kind of information. And so usually in most analyses, what people do is they actually partition their data up at the start and leave some of it for the end, so that they've got a fresh data set to test whether it really generalizes.
I think the thing that I'd add to that is that there's also this question of explainability. And so being able to try and understand or to explain what features your model has learned is really important in order to check that it's not something that you're actually not interested in. So for example, with brain MRI, we often have issues with people moving in the scanner. And sometimes patients might move more if they have a particular outcome which you're trying to predict. And so those things can be confounds that you're actually not interested in training on motion, because why go to the expense of paying for an MRI scan if you could just look to see whether or not someone's moving. So I think there, again, even if you are then sort of trying to validate in a different data set, you might still have the same problem. I mean, this is similar to some of the work in machine learning where people were trying to look at, I can't remember the data, the exact question, but kind of looking at wolves and seeing whether or not they, but what it was classifying on was whether or not they had snow behind them. And so I think that comes back to this question of explainability and trying to understand exactly what your algorithm has learned and whether or not that's something that's acceptable to you or something that you're not interested in.
Cool, thank you.
Well, I think that's a great place to stop, if that's okay. I thought we've had a really good explanatory finish, as it were, to some of the principles underlying these techniques, and a great set of discussions and terrific questions from the audience. Thank you so much for joining in at the end. I hope you've enjoyed your evening. I hope you've really enjoyed our three cutting-edge speakers, and like to show the appreciation as we usually do on these occasions.